benefit from a specific type of antiarrhythmic drug therapy. And I would like to highlight that this is really mainly the work of Andy, who is here, and of Fahima, who is here, who put actually quite a few years of hard work and hard brain work into that. That's actually a picture taken at the celebration a week or two after we had the acceptance of the paper that I'll talk to you about. You know that we use antiarrhythmic drugs to maintain sinus rhythm and to restore sinus rhythm in AF patients. They are used more often than catheter ablation, even though catheter ablation is increasingly used. And the main pattern that you see here from the ESC guidelines to guide our choice of antiarrhythmic drug is really to avoid side effects. So we pick the ones that seem safe for a given patient group, and therefore we have less choices in patients with heart failure than in patients without structural heart disease, and we have a bit more choices in patients with coronary artery disease and left ventricular hypertrophy. And that is all good, and I think it's important because the first rule of not doing harm is relevant, especially when you treat patients to improve their symptoms. But on the other hand, we all know that there are patients who fare remarkably well on antiarrhythmic drug therapy for very long periods of time, whilst others have early recurrences, and you think, well, why did I actually start this therapy? Patient is back after 2 months and already has a recurrence, and we don't really have markers or explanations why that happens. Now, in 2007, at least for my thinking, there was a real game changer, and that was the first report from Iceland that SNPs on chromosome 4q25, so common gene variants that are found in the population close to the PITX2 gene, which is a transcription factor, are better and stronger associated with AF than any other genomic marker. That has since been replicated in many studies in different populations, and there are, for now, at least 3, I think by now there are 4 or 5 independent of these common gene variants in that area that are associated with AF. The rare allele of the most common of these variants, so the first variant that was identified, has been studied in small rhythm control, or small to medium-sized rhythm control patient groups, and it is associated with higher recurrence of AF after all types of rhythm control interventions that you can imagine. Now, PITX2 is sort of the gene that comes to mind when you look at these SNPs, even though it's important to mention that the SNPs are actually a few kilobases upstream of the actual PITX2 gene. PITX2 is a transcription factor that is relevant for embryonic development of left-right patterning, for example, for the outflow tract, for parts of the left atrium, for the sinus node. But it's also interestingly expressed in the left atrium in patients and in mice. And it's almost exclusively expressed in the left atrium. So if you were a stem cell researcher who wanted to design a protocol to develop left atrial cells, you would look for PITX2 positive cells probably more than for anything else. Is this relevant in patients undergoing ablation therapy? This is a cohort of patients who underwent surgical AF ablation and therefore had excision of the left atrial appendage so that we could measure left atrial PITX2 mRNA. And yes, there is a differentiation. So there is a gradient of PITX2 expression in those patients, and you can identify, shown here in red, a group that have low PITX2 levels in the left atrium and are undergoing rhythm control therapy. Why is this relevant? And this is really the sort of slightly unexpected observation that triggered this whole study. We tested the effect of the sodium channel blocker flecainide in wild-type mice and in a mouse model that has a reduced PITX2 expression. And you see that flecainide suppresses inducible AF a bit in wild-type mice, but completely suppresses inducible AF in the PITX2 deficient mice, and that difference is statistically significant at this level, so that PITX2 deficient left atria respond better to flecainide therapy. We tried to understand why that is the case and actually came back to something that relates to my early research days when I was a research trainee. It appears that flecainide induces post-repolarization refractoriness, and that's one of the mechanisms by which sodium channel blockers are believed to prevent arrhythmias. And it does that much more effectively, shown down here, in PITX2 deficient left atria. This blue line shows you APD 90, and the gray line shows you the earliest excitable moment at the action potential, and these are all mean values taken from the study. And you can see that the distance between these two, so the post-repolarization refractoriness, is much bigger in the PITX2 deficient atria than in the wild-type atria. We then looked at the cellular electrical dysfunction in those mice. We have published before that there aren't many structural differences, at least not at the microscopic level that one can see. And one of the main changes that we found is that the PITX2 deficient left atria had a slightly depolarized resting membrane potential. The other difference, and that again has been published by us and by others, is that they have a shorter action potential duration. It is actually a phenotype that is not dissimilar to cardiomyocytes that have a slight metabolic imbalance, interestingly. We then took cells from atria with normal and reduced PITX2 levels and put them through a protocol where we control the resting membrane potential in the experiment. And if you do that, you don't find any electrical differences anymore between the cells. So the difference in resting membrane potential seems to drive the main electrical differences that we observe. What you see here is the inhibition of the sodium current in both genotypes, and you can see that these measurements are identical. And we then did the same thing in human cells, in HEX cells that express the human cardiac sodium channel, and found the same thing, that if you depolarize the resting membrane potential, you inhibit the sodium channel. We were then looking why the resting membrane potential is actually different in those mice. And the first candidate is IK1, and this slide shows you that there is no difference in IK1, but there is a difference in the barium-sensitive background currents in those two mice. And those of you who are cellular EP experts can think about what type of currents those may be. The ones that came to our minds are the task currents, and the reason why they came to our minds is because we did mRNA and protein expression measurements that actually identified a difference in protein expression in those cells. I don't want to go into the experimental details, but want to end with this summary figure. You know that Jack forces you to produce a summary figure, which is probably a smart thing to do. So we had to do that. So if you have low PITX2 levels, and these are sort of the measurement dots in the patients, you're probably prone to AF because of the shorter action potential and the depolarized resting membrane potential. At the same time, this electrical alteration will lead to an increased binding of sodium channel blockers to the sodium channels, and thereby generate more effective treatment with these medications in patients. And thereby, if we now had a way to measure atrial PITX2 levels, and there is a challenge there, of course, you could say this is a group of patients that will respond well to flecainide therapy, and this is a group of patients that will not respond well to flecainide therapy. If you have patients from whom you have left atrial tissue samples and who are on flecainide therapy, let us know. We have a real interest because that is sort of the next step forwards to prove this hypothesis. Thank you for your attention.